microbial genetics. Today, specifically, I'll be teaching you guys a bit about DNA replication and transcription. So as we just said, I'll be doing some bacterial or microbial genetics with you guys, and this has been termed the central dogma of biology. And this central dogma states that a DNA contains the instructions for making a protein, which are copied by RNA. RNA then uses these instructions to make a protein. So if I can just use a little pointer here, so you can see the DNA will be transcribed into RNA, which we'll see over here, and then later the RNA will be translated, or the mRNA, messenger RNA, will be translated to create proteins. So for today's lecture, we'll only be looking at the replication process of how this DNA is copied, and we'll also be looking at how the DNA is converted to RNA. But the take-home message from the central dogma is we move from DNA to RNA to protein. Okay, so first, what is DNA? DNA stands for deoxyribonucleic acid, and it is one of the 4 major types of macromolecules essential for life. They are just coincidentally, the others are proteins, lipids, and complex carbohydrates, also called polysaccharides. DNA carries the genetic instructions for the development, functioning, growth, and even in certain cases, the death of cells. The death of cells is also known as apoptosis. And it is also important for the inheritance of traits. That is how genetic traits are carried from one generation to the next. DNA strands are known as polynucleotides, poly meaning many, so many nucleotides, because they are comprised of simpler monomeric units called nucleotides. So if we look to the structure of DNA, as we just said, DNA comprises of a polymer of nucleotides with the sugar, this is represented over here, being deoxyribose. And then the bases, which are sorted into 2 groups. You get pyrimidines and you get purines. Your pyrimidines are cytosine and thymine, and your purines are adenine and guanine. And then the backbone of the DNA strands, which is represented by this purple or pinkish coloration over here, it has a sugar phosphate backbone. So here, you can see the phosphate group there in the middle. This is your phosphate, and then this is your sugar. And these are bound together by 5-3 phosphodiester bonds. That 5-3 numbering is also important because it gives directionality to your DNA. So this DNA strand over here runs from a 5' to a 3' end. And where those numbers come from is if you number the carbons in your sugar ring. So we start after the oxygen and you move in a clockwise direction. So that's your first one, second one, third one, fourth one. And then the 5th one is actually not on or in the ring. It's just to the side here. And that's why this is known as the 5' end, because the 5' end is at the top. And if you do it with the bottom sugar and you start numbering again after the oxygen and you move clockwise, you'll see it's first carbon, second carbon, third carbon. This is why this is called the 3' end. So that's why this, if you move from the top to the bottom, this is in the 5' to 3' direction. So DNA is comprised of 2 complementary strands, and the molecule shape is a double helix. And for these 2 strands to be held together, these bases need to be paired. The purine and pyrimidine bases of the complementary strands are paired together. In this, for this to occur, adenine, which is your purine, combines or pairs with pyrimidine by 2 hydrogen bonds, and and guanine, your purine, pairs with cytosine, the pyrimidine, by 3 hydrogen bonds. I'll show you an image now, just illustrating that. So here is a model of a figure depicting what I just said, where you can see the cytosine of the 5' to 3' DNA is bound or paired together with the guanine using these 3 hydrogen bonds. And again, notice how these strands are anti-parallel. The one runs 5' to 3'. Again, this is the carbon, so it's 1, 2, 3, 4, 5. And the other one, so it runs from the 5 to the 3, because here, if you count at the bottom, it's 1, 2, 3. There's your third one. So it's from the 5' to the 3', and the other one is the inverse of that. because again, if you number them starting after the oxygen, counting clockwise, it's 1, 2, 3, that's your third carbon. And the same here, counting clockwise, 1, 2, 3, 4, 5. So this is your 5' direction. Also important to note that these these phosphodiester bonds that hold these phosphate groups and sugars together are covalent bonds. So they're bound more strongly than these bonds over here. So DNA, just to summarize, it's a polymer of nucleotides, and the bases, again, adenine, thymine, cytosine, guanine. It has a double helix structure associated with the proteins, and the backbone is deoxyribose phosphate. And this here you can see again the backbone of both sides, and then the base pairing in the middle, which are held together. And this is what gives the DNA its structure. These hydrogen bonds between the AT, so that's adenine, thymine, cytosine, guanine. These hydrogen bonds are what bind these two strands together, and the strands are anti-parallel, the one running 5 to 3 and the other one 3 to 5. Just as an interesting note, if you look at this image here, so this is actually a X-ray diffraction image that was captured by Raymond Gosling, who was a postgraduate student of Rosalind Franklin. Now, this image was used by Watson and Crick in the development of their double helix model for DNA, but there was some controversy there because Rosalind Franklin didn't get a lot of credit for her contribution, although Watson and Crick went on to win the Nobel Prize. So moving on to DNA replication. So DNA replication is a complex process involving numerous proteins to help ensure the accuracy. The first step in this for replication to occur is that the two strands, the two parental strands, you can see over here on the image, need to separate, because both of these end up serving as a template for a new strand. Here you can see the parental strands are these orange with the orange lines, and the daughter strands are the red and darker blue being added, and these parental strands are used as templates. So each of these strands will end up serving as a template for a new daughter strand that's forming, and then once this replication is complete and cell division is complete, one of these parental DNA, one strand will end up in one daughter cell and the other one will end up in another daughter cell in what is termed a semi-conservative model. And this essentially, so DNA synthesis is semi-conservative, meaning each daughter cell obtains one old and one new strand. Let's look at some patterns of DNA synthesis. So in bacterial DNA is different from eukaryote DNA in that most of it's circular in most instances. This means that although it has a single origin of replication, replication will be bidirectional. So from this origin that you can see represented by this green square, replication will happen in that direction and in that direction. So there will be two replication forks that form. You can see over here. With a replication fork is where the DNA, the original template strand, is unwound and then serves as a template for replication. So there will be two of these forks which will continue on throughout the circular DNA until a termination site is reached, where replication will then end and these two circular DNA strands will then separate. Here you can actually see a micrograph of an E. coli chromosome during replication, and there you can see is one replication fork, and here you can see the other one, and these will continue on until the entire circular DNA has replicated. So looking at the components of a replication fork, there's a bit of terminology that I'm going to have to run through with you guys here. The first being that helicase, which you can see on the end over here by this. There's the term showing helicase over there. Helicase, you can almost think of it as a zipper that's unzipping a zip. It's a enzyme. So in biology, most of the time when a word ends in ase, you can check to see if it's an enzyme, because most of the time it will be an enzyme. So that's just a little tip for you guys. So helicase acts as a zipper that unzips and separates the parental DNA strands. And then polymerase, which you can see there's polymerase 3 over there and polymerase 1. Polymerase, you can almost think of it as a builder molecule. So again, it's an enzyme, but it's responsible for catalyzing the addition of nucleotide substrates to DNA in the 5' to 3' direction during DNA replication. And it also performs proofreading and error correction. And please note that there are multiple types of DNA polymerase. Here are just 2 are illustrated here, but there are others. But something that's important to note about this DNA polymerase is that fact that it can only add DNA in the 5' to 3' direction, because this creates a difference in the 2 strands, the 2 new strands. So you can see here at the bottom or at the top what's called the leading strand. And this is called the leading strand because the DNA polymerase can attach to this molecule, and then its directionality, because it runs from a 5' to 3' direction, you can just continuously add new nucleotides to the daughter strand. So it can attach once and just move along. And as this parental strand unwinds, this polymerase at the top can just keep walking along the strand with completely uninterrupted. On the other strand, remember that they're anti-parallel. The directionality does not allow for that, because here the polymerase will need to be placed down and it'll walk a portion. But now, because it was placed down, for example, over here, as this DNA starts to unwind, more and more gets exposed. So it needs to continuously be moved up the new daughter strand, or up the strand, so it can create a new daughter strand. And to do this, it requires primers, because these primers essentially tell the polymerase where to start and where to act on, where to start replication. And that's why the bottom strand is also known as the lagging strand, because it results in these disconnected, small, fragmented portions of replicated DNA. That's also called Okazaki fragments. So Okazaki fragments are just short sequences of DNA nucleotides that are synthesized discontinuously, and it's discontinuous again because of the directionality, where polymerase can't be placed down once and just move. It's placed down once, does a little bit, must be placed up higher upstream and does a little bit more, and then placed again, does a little bit more. So it creates these short little disconnected segments of DNA. So for these segments of DNA to become one continuous strand, it must obviously be connected. And the enzyme responsible for this is DNA ligase. So you can almost think of it almost as a little glue enzyme, where this enzyme, represented by this yellow structure here, moves along the lagging strand of DNA, and it connects these Okazaki fragments by forming a bond between the phosphate group on the one strand and the deoxyribose group of another. So yeah, it just moves and connects these little fragments together, forming a continuous strand of DNA. Some other factor of other definitions I just want to provide you with: DNA gyrase, which you can see here at the start of the replication fork. This is also the ACE kind of gives away that it's an enzyme that catalyzes the ATP dependent negative supercoiling of the double-stranded closed circular DNA. This just aids in maintaining optimal structure for replication to occur, so that the DNA doesn't remain tightly wound together and bundled up. And then the SSB is also an interesting thing that I want to note for you guys. These are single-stranded binding proteins, and these are found in bacteria such as E. coli. And after the parental strand of DNA has separated, these binding proteins bind to the separated strands. You can see them down there and up there. And their function is simply to keep these strands apart so that they don't become just connected again to each other, so they don't pair off immediately after separation. Their job is simply to keep them separate. And they also aid in maintaining DNA metabolism during replication, repair, and recombination. And then just to provide you also with the formal definition of a primer. So our RNA primer is simply a short DNA or RNA segment, and again, this is added by primase, and this primase gives the instructions for the polymerase of where to start adding, where to start building. So they're almost like instruction givers. You can almost view them as coordinators in a way. So the dynamics of this replication fork. So firstly, DNA A proteins bind to the REC, the origin of replication. And this DNA A protein is a protein that activates the initiation of DNA replication in bacteria. And it causes these strands to bend and to start separating. So following this, DNA B and other helicase, remember I said helicase is the zipper enzyme. They separate the strands, and those SSB, those proteins attached to keep these strands apart, the single strand DNA binding proteins, the SSB, they attach to these strands to make sure that they stay apart, that the two strands, once they're separated, that they stay apart. And then the lagging and the leading strands are synthesized. Remember, lagging strand, these small fragments that are then connected by ligase, and the leading strand is the continuous movement. And then DNA polymerase 1 removes the RNA primers and fills the gaps with DNA. And then the Okazaki fragments again are joined by ligase, the glue enzyme. Here are just some formal definitions for you guys regarding the proteins and the functions that these proteins have in the replication machinery. I'll be discussing a few of these in more detail now. I just think this table is pretty helpful for you guys to study up on the formal definitions and what these do. So, but we will be discussing a few of these in more detail now. So, for example, we will be talking about DNA polymerase. Reminder that this is the building protein. This is the one that's responsible for adding new nucleotides and building the daughter strand or the newly synthesized strand of DNA. And it also synthesizes solely in the 5' to 3' direction. And for this enzyme to function, it requires a template, and this template will be the parental strand of DNA. Both strands of the parental strand of DNA will act as template during DNA replication. And then a primer added by the primase. This is a DNA or RNA strand, which essentially just tells the polymerase where to start the building process. So again, on the leading strand, there will be a single primer. On the lagging strand, there will be multiple primers because it'll need to be moved up manually to overcome the shortcoming that it can only synthesize in the 5' to 3' direction. And then it also, of course, needs DNTPs. So DNTPs are deoxyribonucleic triphosphate, and these are the phosphate group, the deoxyribose sugar, and then the nitrogen base. Again, these bases are separated into two groups, the purines and the pyrimidines. So those are your adenine, guanine, cytosine, and thymine. Looking at the polymerase, so polymerase 3 specifically. So if you look at E. coli and bacteria, typically have 5 DNA polymerases, very creatively termed DNA polymerase 1, 2, 3, 4, and 5. This is slightly different in eukaryotic cells because eukaryotic cells also have 5 DNA polymerases, but they're numbered using or identified using Greek alphabet letters, not numbers. So if you look at the DNA polymerase 3 holoenzyme, it is a complex of 10 proteins. So you can see over here, this is where all your proteins are numbered, and there are 10 of them total. Hence, it comprises this enzyme consists of 10 proteins, and it has 3 functional components. You can see there's the clamp at the top, the core polymerase, and then the gamma complex here at the bottom. This Y is a small gamma. And then something that you might note is that this tau, or looks like a little T term, doesn't fall into any of these core or functional components, and that's because its sole function is to simply bind the gamma complex to the core polymerase and the clamp. So it's only a little binding protein. And then the two core enzymes, there are 2 core enzymes present in each polymerase, and they catalyze DNA synthesis. That's the first function. And then the second function is that it proofreads the strands for fidelity to make sure that no mistakes are occurring, because again, these will later serve, if there are mistakes in your DNA sequence, this can result in problems regarding protein synthesis where the wrong protein is synthesized or no protein is synthesized. Some other replisomes, again, just as a reminder, helicase, this is your zipper enzyme. So this is the one that moves and breaks all the hydrogen bonds between the two parental strands of DNA at the start of the replication fork. Your single-stranded binding proteins, your SSB, these keeps the strand apart for the replication to occur. And now the one that I always have to think about before I say, the topoisomerases. Now these break one strand of DNA to relieve the tension from the rapid unwinding of the double helix and prevent supercoiling. Again, during replication, this is important because you don't want your DNA to be all clumped up and together. You want the strands to be nice and separate so that the enzymes can move or walk through the strands unimpeded. Going on to DNA gyrase, this is an example of a type of topoisomerase, and like I said, this induces the supercoiling to help, negative supercoiling to help compact the bacterial chromosome. Here you can see the DNA gyrase represented on this diagram. And then primase, I mentioned these before. Primase adds, they synthesize the short complementary strands of RNA. So here you can see the DNA primase, and it's going to synthesize these short strands, the primers, on the lagging strand. Note that, so for example, there is one primer, and there is another primer, and here will be another one forming. So you will see that on the lagging strand, there are multiple primers needed because this the polymerase needs constant instructions of that they need to move this way, and then because they'll move up, bind, and then start building until they reach close to this terminus, then they will have to be moved up again and bind and move down, where at the top they bind once and they just move, because this is from, like you said, the 5' to 3' direction that they can synthesize new nucleotides in. So yeah, just again as a reminder, DNA polymerase synthesizes 5' to 3' direction only. The lagging strand has these short Okazaki fragments, and each Okazaki fragment requires a new primer. So that's why you will have multiple primers on your lagging strand and only the one on your leading strand. And then these fragments need to be linked. So these Okazaki fragments, and this entire slide, all of this is only pertinent to the lagging strand during DNA replication. And these strands, these small little pieces of DNA now need to be linked together to be converted into one continuous strand of DNA. And this happens by ligase. Ligase is what adds these fragments together. But as you can see by that ATP and NAD plus there, this is an energy intensive process. So it requires either ATP or NAD plus as a cofactor. And notably, something that's just for some extra interest here is that all bacteria contain NAD plus dependent DNA ligase, and the uniqueness of these enzymes to bacteria make them an attractive target for novel antibiotics, because you can effectively target and single out prokaryotic bacteria while leaving other cells like human cells, for example, unharmed. So the bacteria will, or the antibiotic will only kill the bacteria or hamper the bacteria and not normal cell function. Moving on to proofreading. Again, this is important because you need to ensure that the fidelity of these strands are maintained, so that once this DNA starts to code for proteins, that the wrong protein isn't coded for, or that the protein doesn't just become nonsense and doesn't happen at all. So this is carried out by DNA polymerase 3. So it has its own almost form of a spell check, if you want to call it that, built into it. And this DNA polymerase 3 will read the newly added base before adding the next one. And then the polymerase will check whether the newly added base has paired correctly with the base in the template strand. And then if a mistake is found, replication will stop until corrected. So if I could draw your attention to the diagram here. So the purple here represents your template strand, and the blue is the new strand being added. And here at the end, so the newest base pair, you can see there's an A that has been added on the C template, which is incorrect because adenine doesn't pair, cytosine doesn't pair with adenine. This needs to be a G. So this will result in a problem because the hydrogen bonds to bond these two molecules together won't work, because the one requires a 3 hydrogen bond and the other one a 2 hydrogen bond. So as the text here says, it makes an error and inserts the wrong nucleotide, and because these two can't bind, then the strand will not, these two strands won't bind together, and this will cause the DNA polymerase to stop. The wrong or erroneous nucleotide is then removed by exon nuclease activity of the DNA polymerase as it chews back. So it'll stop what it was doing and move back to the 5' end, and then cleave out this wrong nucleotide. And then once the wrong one has been removed, it'll just simply keep going and add new nucleotides. And now you can see it's been corrected. Now there is a guanine that's to pair with the cytosine, so the mistake has been corrected. It's important to note that this activity is not 100% efficient. Mistakes are still possible. So if we move on to some other proofreading, as I said, some errors are not caught or corrected during replication. If this error is in fact caught after replication, it is known as mismatch repair. So if it is corrected after the replication has ceased, it is known as mismatch repair. Here's an example down here of a nucleotide excision pair, where again so here the blue represents your parental strand and the green your daughter strands, and again next to the guanine a thymine has been added, and this is wrong because this needs to be a cytosine. So then this is corrected, and the cytosine, the T is cleaved out and the cytosine is added to correct the mistake. So the termination of replication is important because that's obviously how this will stop. But in circular DNA, this presents a new challenge because of the structure of the circular DNA. So the replication will stop when the replisome reaches a termination or TER site on the DNA, and then catenanes form when the two daughter chromosomes do not separate. So a catenane is just a mechanically interlocked molecular architecture consisting of 2 or more interlocked macrocycles. That's a very complex way of saying that these two circular DNA structures are just linked almost together like a chain link. They're hooked into one another. And then, so to solve this problem, because this would mean that if this were to remain like this, the DNA wouldn't be able to separate into separate daughter cells. So to combat this, topoisomerases will temporarily break the DNA. You can see over there, it has broken open that red strand of DNA so that the strands can separate and then move into the respective daughter cells. Moving on to RNA. So RNA also comprises of a polymer of nucleotides, but there are differences between RNA and DNA that you should definitely be aware of and know. So the first difference is that the sugar in RNA is no longer deoxyribose, it is now only ribose. And here you can see in picture A, it's the difference. This is deoxyribose, this is found in DNA, and then this is ribose, which is found in RNA. So there is a difference in the sugar. And then the other important or main difference is that the bases are slightly different. So RNA still contains adenine, guanine, and cytosine, but it does not contain thymine. You can see here in figure B or image B, thymine is found only in DNA, and it gets replaced with uracil or U in RNA. So if you see U on a strand of DNA, you can be sure it is RNA. But there are some similarities too. The phosphodiester bonds on the backbone of the molecule remains the same as it was in DNA. Also, another structural difference, most RNA molecules are single-stranded. For you kids interested in microbiology or hoping to continue on, you will eventually start dealing with RNA that's also double-stranded. For example, certain viruses have double-stranded RNA. The rotavirus is a is a good example. It's the most common cause of acute gastroenteritis in infants and young children worldwide, and it has double-stranded RNA. And then there are three different types of RNA that will differ in from each other in function, cytosynthesis, and structure. This is messenger RNA, mRNA, ribosomal RNA or rRNA, or and then transfer RNA or tRNA. We will be discussing these in more depth later in this lecture and in the next one. So transcription, moving or continuing on with starting with the transcription of RNA. So this is the synthesis or the creation of RNA using DNA as a template. And so the RNA that is produced from this will use one of the two strands of DNA as a template, and it will have a complementary sequence to the template DNA. Interestingly, so that means that the RNA that is produced from a template DNA, a template strand of DNA, will have the exact same base pairing as the non-template strand of DNA, with the exceptions being the sugar will be different, and then thymine will be replaced with uracil. There's a very good picture later in the lecture that I'll more clearly illustrate this for you guys. And then, as I mentioned, the three types of RNA. Messenger RNA carries the message for protein synthesis. Transfer RNA, which you can see down here on the right, it carries amino acids during protein synthesis. So here you can see it's the nucleotide pairs on it, and this where this blue dot is, this is where the amino acid will be attached to the tRNA molecule, so it can transfer these proteins. We'll go into more detail during my next lecture on translation, but this is just to give you an idea of what that molecule looks like. And then ribosomal RNA or rRNA, they are the components of ribosomes, which are also important during protein synthesis. We'll get there. So during transcription, a DNA sequence is copied to make RNA, and this is performed by an enzyme called RNA polymerases. And it's controlled separately for each gene in the genome. And then transcription involves three stages: initiation, elongation, and termination. In the next couple of slides, we'll be discussing what this RNA polymerases looks like and what exactly it does. We'll be talking a little bit about gene control, and then I'll explain each of these three stages to you guys. So RNA polymerases, here's what that molecule looks like. As was the case with polymerase that we discussed earlier, it is the builder protein. It's an enzyme that synthesizes the RNA from the DNA template during the process of transcription. So during this process, the information stored in the molecule of DNA is copied into a new molecule of messenger or mRNA. From this picture, you can see that RNA polymerase is a complex molecule composed of protein subunits. And so here you can see there's different subunits are in the different colors and the different Greek letters. That represents all the different protein subunits that make up this RNA polymerase molecule. It also uses the enzyme helicase, the zipper enzyme, to open the double-stranded DNA so that one strand of the exposed nucleotides on the DNA can be used as a template for synthesis. Important difference here between prokaryotes and eukaryotes. So bacteria and archaea contain a single type of RNA polymerase, while eukaryotes contain three distinct types, each responsible for the synthesis of a distinct subset of RNA. So for you guys, really important just to know that bacteria and archaea only have a single type of RNA polymerase. Moving on to promoters. So I think let's start with the definition here. A promoter is simply a sequence of DNA to which a proteins bind that initiate the transcription of RNA. So essentially, a promoter is important for transcription initiation, as it tells the polymerase where to sit down on the DNA and begin transcribing. And so if we look at this table over here that reads some of the alternative sigma factors in E. coli and their cognate promoter sequences, I thought it's just important to give you a bit of a clarification regarding that cognate word, because it is a term that is used quite frequently when it comes to microbial genetics. So cognate is a term we borrow from linguistics that signify correspondence or corresponding. So you can also read this as alternative sigma factors in E. coli and their corresponding promoter sequences. And so what are these alternative sigma factors? They enable bacteria to change the promoter specificity of the core RNA polymerase to enable the expression of genes that give them advantages in particular situations. It's a real mouthful, but it essentially just boils down to, if there is some form of stress, if you look at this pink or purple portion of this diagram down here, so starvation is now a threat. It's enacting on this molecule or the cell. So now the cell can use its sigma factors to increase the expression of, you can look at it as increasing the expression of genes that will help it to survive starvation. So it can be a response to environmental stress. So you can look here where it says heat shock responses. So if there's a heat shock, the E. coli is able to express these genes, these sigma factors to help it to deal with the shock response. You can also look at chemotaxis, which is the movement of the organism into or away from the concentration of a chemical or motility and flagellar components. All of this deals just moving the organism. So there are some important things I just want to tell you here also, is that prokaryotes only carry 3 promoter elements, the -10, -35, and UP or up elements, where eukaryotes carry a wide variety of promoter elements. This difference in the number of promoter sequences allows for transcription in eukaryotes to be more highly regulated. In addition, the use of enhancers in eukaryotic transcription allows promoter sites in remote location on the DNA to initiate transcription. The take-home message from all of that is simply that transcription in eukaryotes can occur with less stimulation than in prokaryotes. So now, transcription initiation. So transcription begins when the RNA polymerase binds to a promoter sequence near the beginning of a gene. So important to note that here, this entire region here is your promoter region, and it'll tell the polymerase where to bind and to start transcribing. So again, just as a reminder, this sequence is important because it tells the polymerase where to sit down on the DNA and where to begin transcribing. So here you can see the transcription. This transcription start site is over here, and that's the direction we're going to move in this example. In bacteria, each group of genes transcribed together has its own promoter consisting of short sequences at the -10 and -35 positions upstream of the transcription start site. So see how this is towards the upstream area of where the actual transcription will take place. So this sequence at the -10 is also called the Pribno box or the -10 element, and usually consists of 6 nucleotides called a hexamer. This hexamer is usually TATAAT. So that's thymine, adenine, thymine, adenine, adenine, thymine. And the function of this sequence is that it's essential to start the transcription. Whereas the other sequence, the -35 sequence, also a hexamer of TTGACA, its presence allows for a high transcription rate, and together they make up your promoter region. So following initiation, we have elongation. So basically, elongation is the stage where the RNA strand gets longer thanks to the addition of new nucleotides. So during elongation, you can see here, this is your RNA polymerase. It is now essentially bound to the DNA template and will now walk or move along one of these strands, along the template strand in the 3' to 5' direction, and it'll add new nucleotides to your RNA. So after binding, the RNA polymerase unwinds the DNA, which happens over there, and a transcription bubble is produced. Note that this transcription bubble just refers to the region of opened up DNA. And then it moves with the polymerases as it transcribes mRNA from the template strand. And within the bubble, there's a temporary hybrid of DNA RNA that is formed. And just, yeah, as a reminder again, this RNA transcript is nearly identical to the non-template strand of DNA, with the exceptions being that uracil is in the place of thymine and it has a different sugar in the nucleotide. So after elongation, we have termination, and this occurs when the core RNA polymerase dissociates from the DNA. DNA sequences mark the end of gene once the polymerase transcribes a sequence of DNA known as a terminator, or that TER that we had earlier in the lecture. I'm not going to go into too much detail about this, but just know that there are two termination strategies found in bacteria. There's a row dependent and a row independent strategies. Looking at what this termination actually looks like. So the RNA polymerase will pause in an A-rich or adenine-rich region of the terminator, and it allows for the formation of a stem loop structure. Here you can see the stem loop forming. And this stem loop destabilizes the uracil adenine bonds that are not able to hold together the DNA RNA hybrid, and thus termination occurs. So here, this is your termination sequence in your DNA. The RNA moved along, hit the termination sequence, and then both the RNA polymerase and the RNA strand are then separated from the DNA strand. So giving an overview, a quick overview of the entire process, so of transcription process, the RNA will bind to the promoter and the gene will start to unwind. So here is your RNA polymerase. That red section on the DNA signifies our promoter region. So it tells the polymerase where to sit down. So it's sitting down on the DNA and it'll now move in that direction. So to the right in this example. And then the RNA is synthesized by complementary base pairing of free nucleotides with the nucleotide bases on the template strand of DNA. So as this polymerase moves, it'll add new nucleotides. So here you can see the RNA nucleotides being added and the template strand serving as instructions for the order of these nucleotides. And here, I think this image illustrates very clearly what I said earlier of how the similarities between RNA and the non-template strand of DNA. So you can see, for example, here, the RNA will be GAC, but look at this non-template strand. That's the one at the top here. It's also GAC. And that's because they have to be the opposite pairs of what's found on the template strand. So the difference being, though, that before the GAC on the RNA, you find this U, this uracil term. On the DNA, that would not be a U, that would be a T or a thymine nucleotide. So as the site of synthesis moves along, the DNA that has been transcribed rewinds, so it takes back its original double helix structure. Now this RNA polymerase will reach the terminator in the next step, which will then separate the RNA polymerase and the newly formed RNA. And the entire double helix of the DNA will be reformed to be used as another template. So I just decided to include a couple of review questions. I'll give you guys maybe about 15 seconds for each of these. This doesn't count any marks. This is just to kind of, for your own benefit, to see how much of the lecture you actually, remembered. Like I said, not for marks or anything. This is just to help you guys study and to see how much you gain from the lecture. So for the first question, DNA carries instructions for which of the following? A, cell development, B, cell function, C, cell growth, D, reproduction, or E, all of the above. Like I said, I'm going to give you about 10 more seconds on this. So the correct answer here would be E, all of the above. Remember, just as an interesting note, that in certain instances it can also carry instructions for apoptosis or cell death. Moving on to review question number 2, which of the following is applicable to DNA? The sugar is a ribose, the bases are adenine, guanine, cytosine, and thymine. The sugar is deoxyribose, the bases are adenine, guanine, cytosine, uracil. The sugar is deoxyribose, the bases are adenine, guanine, cytosine, and thymine. The sugar is a ribose. Bases are adenine, guanine, cytosine, uracil, or E, none of the above. Again, I'm just going to give you 10, 15 seconds. So I hope you guys are done. The correct answer in this one is, of course, C. Remember that an RNA, your sugar is a ribose, and uracil, as seen here and here, are found exclusively in RNA. So that leaves C as your only correct answer. Moving on to review question number 3. During DNA replication, which model correctly describes the DNA received by each daughter cell? The conservative model, the semi-conservative model, the dispersive model, the double helix, or E, none of the above. Again, only going to give you about 10, 15 seconds. So the correct answer here, of course, is the semi-conservative model. Remember in that image how the DNA strands of the parental cell, one of the strands ended up in the one daughter cell and the other one ended up in the other daughter cell. Hence, it's a semi-conservative model. So now for the final question, this is perhaps the most complex one because we did talk about a lot of enzymes today. So which enzyme complex is responsible for proofreading? Is this DNA polymerase 1, DNA polymerase 3, DNA ligase, DNA gyrase, or DNA primase? Again, only about 10, 15 seconds for this. And the correct answer for this one is, of course, DNA polymerase 3. Well, that concludes our lecture today. Thank you guys for your time. I